Hello, everyone. I'm Alicia, and I'm one of the organizers. Let me introduce you Dani Ushizima. She is our first speaker for our tech talk. And Dani Ushizima is the computer vision area leader for the Center of Advanced Mathematics for Energy Research Applications at Lawrence Berkeley National Lab, and a data scientist for the Berkeley Institute for Data Science at UC Berkeley. She is one of my mentors at UC Berkeley, and I'm very happy to introduce her today for our tech talk. Let's welcome Dani Ushizima.
Thank you, Alicia, for the introduction. I'll let people kind of settle. Welcome. Welcome all. Okay, so imagine inspecting 1 terabyte of data a minute. And it doesn't matter how good you are at binge watching. We're talking approximately 200 Netflix movies. It's just too much data to sift through manually. But this number of images is actually the amount of data that several scientists that depend on experimental data recorded as pictures need to deal with. Several laboratories here in Berkeley, for example. So what we need is to construct computer programs that help us sift through this amount of data. So that's what I do. I'm Dani Ushizima. I'm a data scientist at BIDS. Also, I'm a staff scientist at the Lawrence Berkeley National Laboratory. And here's a picture of the lab, specifically the Computational Research Division. And for those that don't know Berkeley Lab, it's a big Department of Energy laboratory, which is just up the hill. It's open, non-classified, science-centric laboratory, which worries about everything from biology to materials to groundwater flow, which might be the topic of a work colleague, Deb Agrawal. It also works in gene sequencing using supercomputers, which might be the topic of another colleague, Kirsten Fechner. Now you may wonder, in a big laboratory, there are so many images being collected all the time, and not only pictures of faces and streets, but scientific images to peer inside the materials, to look at cells, to check for oil spills from satellite data. And all of those pictures are nothing but matrices of pixels. And I'm in the business of converting those pictures into information, which means using computers to extract structure, find objects, recognize patterns. So what we need to build algorithms. And one of the places to catalyze this work has been BIDS. So part of my work at BIDS has been to start an initiative focusing on image analysis and image processing across domains, or ImageXD for short. And our goal within BIDS has been to actually increase the awareness of the impact that analyzing and understanding pictures can have in a variety of fields. And what's interesting, by breaking the barrier of expertise, we broke lots of barriers. One of them is the gender gap. It's a very diverse group of people. Besides this initiative that's a year-long work, we also have events, and we divide these events in 3 days in which we learn and discuss, and finally we go coding together. Oftentimes we actually have the chance to code with people that develop the packages that we love so much, such as Scikit-Image, Scikit-Learn, or Matplotlib. Okay, the common theme here is to automate the process of converting images into information and give answers to scientific questions. So let's see some use cases. So I'll talk about a few of them. Can you hear me with this microphone? Great. And these use cases, what they have in common is users. We're working with domain scientists, then with the instruments that they use to capture information about what's going on in their samples, to finally, what's the algorithm that we're helping them with. Okay, first part, So here I'm going to focus a lot on the work that I currently develop with the other colleagues at Berkeley Lab, particularly in the interface between the Computational Research Division, where I live, and the Advanced Light Source, the particle accelerator. Here using a specific modality called micro-CT, which is a type of computed tomography. So one of the problems there, here the users are both academics here from UC Berkeley as well as industry, and they're all interested in this new material called ceramic matrix composite. It's a combination, as I'm showing here, of this orange part with the ceramic matrix with the silicon carbide fibers, which I'm showing in white. Why do they care? They can build incredibly resistant aircrafts, turbines, and other parts for aviation. What's my job together with these experts? To separate different information here, in this case, how you separate the ceramic from the fibers, how you detect these fibers, how you characterize the coating of these fibers. Currently, there are different types of coating. One of them is boronitride, and different thickness of this coating. And also, can you tell anything about the strength of this material based on some of the deformations that I'm showing in green? So this is the final result after processing a lot this raw information that came from the particle accelerator. Another type of sample here is some of the material that NASA is interested in called carbon textiles. And here, this is a protection to the aircraft that is going to missions such as Mars 2020, which is this one. So in order to protect the rocket, they have this wrapping. And their question is, can you give me more information about how this carbon textile is organized? Can you pull apart the weft and the warp part of this textile? So here we use an assortment of algorithms to do transformation of these images into this gray and blue part called segmentation methods. Some of the techniques behind are machine learning algorithms such as statistical region merging is one of them, as well as deep learning algorithms such as convolutional neural networks. Yet another problem, and here all using the same instrument at the advanced light source, microtomography. The question is that the civil engineers here on campus have, well, it seems that these archaeological samples that are part of buildings, these buildings are still withstanding. Is there any secret that we lost through time? Is there something that I can learn from these archaeological samples that I could use in construction today so that we're not rebuilding our bridges every 7 years, right? And the answer is yes. It seems that there's something in there, in the way that these materials were composed together, these aggregates are put together, that made them so special. They can self-heal. So very exciting information that's coming out of these samples. Also, some of the methods, algorithms behind are segmentation to separate the different aggregates and the relationship between these different aggregates within the material. Okay, the past slide was mostly about 3D image data sets and things that probably if we go through and we have a small data set, we could make sense. But what about very abstract patterns, such as those also collected at the advanced light source, but they are like this. They're actually scattering patterns that come from shooting X-ray to different polymers, different materials, metals. And just looking at that thing that looks like a bad rainbow, you infer that, well, this is a good polymer, this is a good film, high quality, bad quality, this is BCP, HCP, so on and so forth. So that's something that that expert, Alex Haxmer, just looked at. Well, I know what it is. I know this is BCP. That's the crystal lattice associated with this particular polymer. I couldn't tell, to save my life. Well, in order to deal with large amounts of data and not keeping that scientist as part of the loop throughout the whole process, we invented methods to classify those scattering patterns into different classes. And I'll tell you a little bit about that story. So that's how it happens. Here is the X-ray heating the sample. there is a scattering pattern that comes out of it. So think Fourier transform, what exactly is happening at that interface of the X-ray with the film. And what we want is to extract the structural fingerprint, how we transform that scattering pattern abstract into a feature vector that allows us to sift through large databases and be able to classify samples that we don't know a priori. So we invented a tool. So this is built here in Berkeley by our teams. It's called PyCyber. It stands for Content-Based Image Retrieval, which means that you ask your database, not with metadata, although you could. Instead, you ask questions with pictures. So given this picture, can you return a line here with other pictures that looks just like that? And that's exactly what this tool does. So there is a user interface that allows you to quickly sift through very large data sets. In this case, for the GSEX, which stands for grazing, incidence, small angle, X-ray scattering, we have about 4 million images, and we can sift through that really quickly. What's behind this looking to images very quickly? What we do, we transform this picture through several layers here, called the convolutional layers as well as max pool layers, to finally get our feature vector. And that's the signature or fingerprint of these images. There's yet another architecture to do this transformation of the picture into a feature vector. This is called Inception ResNet v2, which is a sort of combination of technology from Google and a Microsoft. Long story short, this is a deep convolutional neural network that allows you to recover these feature vectors and sift through very large databases. Here I'm illustrating with these scattering patterns, but pretty soon in the talk you'll see that you can actually use that for other purposes. And here are some of the other purposes. So in collaboration with several people, I've been actually asking myself, can we actually transfer some of the things that we learn at Berkeley Lab, looking at materials, to problems such as image analysis for healthcare improvement? So let me present you some of the use cases. And also having in mind, who cares about women's illnesses? So I was wondering, and I met people that cared about women's illnesses. Some of them are Minna Bissell and Candace Tanner from the Bioscience Division at Berkeley Lab. And the question that they have was, well, I have a series of confocal microscopy of cells undergoing mitosis. And the science question they had for me is, Dan, calculate how quickly this mitosis is happening. Well, I also want you to tell me about the angular velocity. It seems that they're kind of going around. And is it any different, the normal cell from the abnormal cell, when I have these cells in microenvironments? Just using, remember some of the talk of Daphne a little while ago. So how we get these models, we test in mouse, and sometimes we cannot transfer. What's very exciting that Minna Bissell has been doing her whole career is to create these human models. So these are human cells that they develop not only in Petri dish, so it would be 2D, but these 3D models, which means cells that are created artificially, proliferated artificially, but are human cells. So very exciting work. And the job of, here again, segmentation. Things that we use for detecting those fibers, remember, can also be used with some repurposing to segment the different cells. These rods are artificial. This is part of my job of computer graphics, whereas the centroid of each one of these cells, how they are moving. Another project here in collaboration with pathologists, physicists, and engineers. So the question was, can we actually automate cervical cancer? Can we count cell? Simple as that, using this as the input. In the next slides, I'll tell you more about this story. It's a very exciting story. And the role that BIDS played in basically saving this project from dying at the start. And last but not least, Greenberg's laboratory at UCSF has been interested in working with a multimodal, so different kinds of instruments. While this one is confocal microscopy, light microscopy, that one has histology information on light microscopy, MRI, CT, how you put all those pictures together. So one of the things that we've been doing is working with them in registration methods, which are a bunch of matrices, transformations, if you will, of these images so that you can put all of them together as part of the understanding how Alzheimer's starts, how we're going to find the cure, what kind of drugs should we be testing. And that's one of the works that Leah Greenberg lead at UCSF, and I had the chance to participate in the group helping testing some of these algorithms in these methods. Another thing that is very interesting about these women witnesses is some of these stats here. 99% of the patients with breast cancer are women. Yeah, male can get breast cancer, but it's really a tiny number. Cervical cancer, 100%. This is really women's disease. In Alzheimer's, which was sort of eye-opening for me. I didn't actually know that women are more likely to have Alzheimer's than men. Okay, let's drill into cervical cancer. If you want a book, and I'm not going to have all that time, take a look at Women's Disease by Elena Lowy. Great book. She's a wonderful author. I really recommend. But in a nutshell, what's cervical cancer? Well, first of all, you need to know where the cervix, I bet every one of you know where the cervix is. It is at the bottom of the uterus, and that particular linen that protects the bottom of the uterus can be infected by HPV. That's not the only cause of cervical cancer, but it's one of them. And HPV, it's not HIV. It's really hard when you pronounce that really quickly. People don't know for sure. HPV is the human papillomavirus, and it's quite interesting. HPV is a ghost that lives in men and does boo in women, right? Does nothing to men, but causes cervical cancer in women. And here's the situation. It's the second leading cause of women death between 20 and 40 years old. More than 500,000 new cases per year. And it kills a lot. It's actually the first leading cause in several countries in the world, particularly the developing world. The thing is, the chances to be cured are 100% if caught in early stages. So that's been part of one of my dreams, is that why not we can catch that early on? Why don't we have that for all women? That's my dream. How does it happen? How we can catch it early? One of the methods is pap smear test using conventional pap smear, and that's what I'm showing here. How we start? Scrape with a spatula, scratch with a brush, put on a glass slide, stain, digitize. Perhaps some of you went through this process. I did. Yeah, no anesthesia. We want that to be highly accurate, folks. We must have it. My interest in cervical cancer really hyped up when I got exposed to the IEEE International Symposium on Biomedical Imaging in 2013 for the conference that happened in 2014. They had this challenge. They put a public data set out there and said, well, can you count the cells? Can you separate where is one or the other? So these are some of the pictures that they made available. So this is close to Christmas. I'm here with a postdoc. She's going to spend Christmas with me. Like, great, we're going to have a party. We're going to be coding. So that was it. We spent Christmas doing that, a little bit of January as well. And we have a very large data set, lots of these pictures just like that one, almost 1,000 images to go through. So we knew that we were not doing that manually. We didn't have that kind of time. Well, after a month and a half, we came up with an algorithm, and these are some of the results of that algorithm. You know, we did it really quickly, and we were super afraid. And my postdoc, Dani, you're crazy. It's not our business. We're going to get last. Like, we got to take a risk. Let's get into the challenge and see what happens. So just to give you a few what's going on within this algorithm, transformation of matrices, right? First, we're enhancing borders with bilateral filters, which is a kind of anisotropic diffusion filter, edge preserving, to then go to statistical region merging and using probably biology from lower school. Well, what's the cell? Cell is a nucleus and cytoplasm. We find the nucleus, cytoplasm is whatever is around it, right? So that was the rationale that we had by them, and we couldn't explore much more than that in a month and a half. So we found the nucleus, got some border around the nucleus. We're calling those intensities typical of the cytoplasm. Find the cytoplasm body, but how we split? Well, as computer science, we do distance. Whatever is closer to one nucleus belongs to that one, and so on and so forth. And what we thought would be the last was actually the first place in the code competition. We were so excited. Like, okay, we're gonna get rich, right? Like, no, we don't wanna get rich. We want that for free for all women. Okay, so at least we're gonna get money. And we started sending proposals, right and left, and no money. Everybody said, no, out of scope. Conventional pap smear, I mean, you're crazy. We're using liquid-based right now. I mean, this is not gonna work. We're not gonna get anyone to pay for this idea. And that's when the Berkeley Institute for Data Science basically saved this project. And it took from this very rough approximation of where are the cells to something much better just in the following year. Not only that, we Pythonized the whole software stack for dealing with that cell counting. Lots of support from Moore's Law Foundation. We also got support from Microsoft to do all the computation. We have a whole team spread all over the world that were basically using Azure as part of the computation. And we went from very rough to classification of abnormal cells, not only abnormal cells, but the different stages of cervical cancer. So very, very exciting. What's the idea behind? Very similar to Pinboard. And I alluded to that when I was presenting those abstract patterns. What we want to find pictures and retrieve others that are just similar as we would do at Pinboard. Well, that's exactly the idea behind PyCyber, the software that we've been constructing. We can use for X-ray, we can use for cells as well. We have an unknown case of a cell. Give me those that match the closest. And that's a starting point. That's at least a pre-screening tool. So that's what we're doing. It doesn't matter the instrument, but in this case, the instrument is a microscope. Actually, there is a scanner that goes through several of those slides at a time. You have digitized cells and an inferential engine that comes from PyCyber that allows you to do this classification. And not only X-ray, but also you can use that for other purposes, such as recognition of vehicles, using with the geospatial data. And here's just an example you can use to query just pictures or actually pictures of your drawing. So your picture doesn't need to leave in the database at all. Okay, this is coming to an end. And to end here, I would like to just to share with you and that's really my personal opinion, when a woman enters data science, it really changed the woman, but when women enters data science, it changed the women. So look to your right, look to your left. Let's build a sorority. Let's change data science for better. And well, with that, I would like to say thank you. You know, all this work would not be possible with wonderful students that come from all over the world to work with us. We have lots of male actually helping with the cervical cancer project. It has been amazing to inspire them also. Like, yeah, I think this is important. You know, 20 to 40 years, if I lose my wife, boy, my family is destroyed. So it has been really, really great. Alicia's one of them, so thank you so much for organizing WiDS and also participating in some of the projects. And with that, I would like to end thanking all the sponsors, collaborators, and I'll be happy to entertain questions if you have any. Thank you. questions? Internships.
It's a wonderful study on how women's issues can be very impactful. Are you making any connections to how to change cultural attitudes about treatment? Because I can see you're going to come up with a solution, but many cultures are really resistant to dealing with those topics, and how do we get the science and the solution to the women?
That's a very hard question on how we're going to change. My personal belief is through education. We need to start really young, and I think that, now that I have the chance, a segue is we need to start with them as young as 5 years old, which is the age of some of those little girls. We need to pass on the importance of a technology, the importance of a health, and how each one of us can change that. But I hear your thoughts. Cervical cancer is a big problem, particularly in the developing world, in places where the society is very patriarchal. HPV is a disease that can be sexually transmitted, and oftentimes like, oh, okay, you have HPV, it's sexually transmitted. You're a sinner, better you die. So this is a very sad outcome of a lot of societies that don't see women as a real human being, a full human being. Any other questions? Okay, I'll stay for lunch and I'll be happy to chat with you. Thank you.